Good morning. You guys remember that I have been talking about pieces of DNA that encode something or that are genes versus other DNA those are not genes. And so today we're going to start understanding what is the difference between DNA that is considered a gene and DNA that is not a gene. A lot of people, whenever they think the DNA on the cells, they're like, oh, that's the chromosomes. Okay, the chromosomes are DNA plus, so there are proteins too. And then they think, oh, that's the stuff that has the information that make us who we are. Like, okay, that's true too, but it's not the whole story. So today you're going to start an annotation exercise. You're going to know what an annotation is. It's basically information on top of information. And one of the things that people do when they do annotation is predict where the genes are in a genomic DNA sequence. Right now, you don't know what these terms mean, but by the end of this lecture, you will know. And when we do a bioinformatics tool called FGenesH, we're going to take one approach to predict where genes are. And when we have our second day of BLAST, that day it will be for predicting where the genes are using a different approach. So there are two approaches for learning where are the genes. One of them is called ab initio, which is like from the beginning, meaning I'm looking at this DNA sequence. I mean, I'm not comparing it to any database of things that I know are genes. I'm just looking at this one sequence and looking at for cues that would say, there might be a gene on this area. And the other one is the homology based, which is I have my DNA sequence and I'm going to compare it to a database of sequences that I know are genes. So there is a big deal in trying to find which parts of the genome are genes, especially when sequencing the genome is more expensive. Or I don't know if you remember Jasmine, the first day she talked about sequencing the exome, which is really looking at which DNA sequences encode the parts that become part of our protein. And this becomes increasingly more common. But you need to know, okay, what do those things mean? So that's what you're learning today. All right, let me share the file with you guys on genome annotation. And I kept saying that there would be gene prediction. Gene prediction is one of the things that people do is genome annotation. For some people, it's the main thing. Other people focus on different types of annotation. But finding genes is some hot area so that we can see if there are some genetic diseases. Most likely, mutations are in the genes. Not always, but if you had to bet, that would be your bet. And that is why people do the whole exome sequencing, or we're migrating toward that because you are looking which parts of the DNA are the genes, and within a gene, which parts are the exons. So you're going to learn about that. As soon as my file shares.